Monday, I don't really do it as a planned lesson as such, but I'm just downloading. I'm waiting on the stuff from my group tuition session, so I thought I'll jump on here. And then if anyone has any questions, I can answer those. Oh, thank you, Apple. That's lovely. Thank you. I'll boot up your laptop. Yeah, definitely do. I'm just setting up. Don't want that. Where's it gone? There we go. Right. That's what I'm doing. What am I doing tonight? You can tell I'm tired. Any tips to revise biology? What is it that you're struggling with the most? Is it the content? Is it the application skills? So the first thing is figuring out what you don't know. So when it comes to revision, make sure you're doing what you don't know. Those topic areas that you hate, basically, start with them. Because that will make a big difference for you. Feeling like my teachers doesn't go into the depths. So if that's the case, then what I would advise is go to my channel and look at the videos that I've posted. So all the videos that I post, it should cover to the depth you need, whether you're an A-star student or whether you are any of those grades right the way down. Everything that's on there is aimed at making it accessible, so it goes into the depth that you need. And if you have questions after watching it, then do please let me know. I use your notes compared to your YouTube channel. No, like, oh no. Yeah, but they might just do it a different way. It's hard. Hamad, welcome. Hi again. How are you? You have a kidney exam tomorrow. Anything to look out for? I don't know. If you've got a kidney exam, the key thing I would say for you is making sure that you are confident in what's happening at which position. Also being able to, so like, for example, there's a question on podocytes in some of the past exam questions, and those podocytes are actually assessing your module 2 knowledge, understanding that cells can differentiate, and knowing about their potency. So just because your tests on kidneys doesn't necessarily mean it won't bring in all the other topic areas. That's what I would say is important, that you try and take a step back and say, right, well, what are they actually asking me? What content am I being assessed on? So that you can then use that to help guide you. So obviously we did last week the different parts of the kidney. I'd say the collecting duct's probably the hardest bit, and it links quite nicely to the hormonal system and also to animal response. Hi, Paul. Any good websites for exam questions? Yes. So AIQ Chat, I forget to plug my own website, but AIQ Chat's a really good one for doing multiple choice questions. Going to OCR website and actually downloading the past papers and doing them, or you can use physics and maths tutors. They've got them in the exam what am I trying to say? Modules. They've got them specifically to the modules. If you go on my website as well, you'll find that I've got some workbooks out there that are not all finished yet. So I need to keep working on them. I'm currently doing the transport implants and photosynthesis, and I'm going to do plant science together. I found the mock papers they're going to use. Should I use it? No. So when you say you found the mock papers that they're going to use, should you use it? I would say no. And the reason I say no is because you're setting yourself up for failure. I know it's an easy way to help you, in a sense, if you are trying to get that predicted grade, and I get it, you want the predicted grade, you want to show the UCAS that you can get the grades that you want. So you think that by accessing these papers, you are going to be able to do well on the paper. But what actually happens is, yes, you may do well in that paper because you've seen it. You may memorize the mark scheme. If your teachers are savvy enough, like I would do, then what I suggest is they're probably going to change those questions around. So you're going to memorize those mark schemes. And I've caught out so many students from doing this. You memorize those mark schemes, you try and answer the question. I've switched it around. And so therefore I catch you out and you're not learning the content, you're not learning how to apply. And so therefore you're not going to benefit and it's going to hurt you rather than help you. So I would say don't look at it if you know what they're doing. And how do you know? Hi, Chidima, I can see you in both places here. How do you know which ones they're going to use? Because if they're using, I mean, 2024 is out there already. So yes, you can see any exam past paper up to 2024 because it's on OCR's website. But that's one of the reasons that the papers get locked down. So that any student who needs an assessment or our teachers who need to set assessments, they shouldn't be available. Stephanie, I don't know. I've literally thought I'm waiting on my group tuition video downloading. So what do you want me to cover? What part of the topic area would you like me to cover? I'm just kind of jumping on because I have some time. That's ridiculous. If your tutor has given you the locked paper, that is really bad of them. That's so unprofessional and that annoys me. Because that's so, one, against the rules of OCR. Two, it is unprofessional of them. They are putting you and others at a disadvantage by doing that. I'm going to breathe. I'm going to either or, Stephanie, I'm going to say to you that they shouldn't be doing that. It's like the worst thing that they can do. And even these people who put out like they want good reviews, but it's not good reviews. They're setting you up for failure because you're going to memorize these mark schemes. You're going to go in and be like, ooh, look at me. I can get whatever grade if you can memorize them that well. But you're not understanding it or knowing it. Ah, you started uni. That's awesome. And I'm sorry, I'm not having a go at you here. I find it so frustrating. Like I've got a student who's got a mock. So I see my student every Wednesday evening and they've got the mock after I see them next week. And I know exactly what papers they're going to be using for their mock. And I will not do those questions with the student because it's not fair for them if I provide a false sense of security. It's really bad. Hi, Maeve, how are we doing? Can we go through transcription practice topic? Yeah, of course we can. Of course we can. Let me jump up and get that. Going back though, Mrs. Lana Ray, how are you feeling about going to university? It's so exciting. I wish it was me, but at the same time, I'm glad it's not. I've had so many students lately who've been sending me messages about either their remarking or about their starting uni. And one of my students was saying that she's still watching my videos, and that made me feel really happy that even at university it is helping with the subject content. So I love that. I'm so excited. I've looked at the course for biology one and it's just exactly, honestly, it really is very similar. That's basically why I loved the first year of uni, because it was like you were doing the advanced part all over again. Tony's just started. I know Tony's just started as well, or Dina profile if you have time. Stephanie, I'll see what I can do. Hold on, give me a second. I am bringing it in now. I'm chatting away, but I'm bringing it in. Don't worry. I will go through. I've got some of my old pictures here. I'm updating a lot of stuff as I go. Oh, thank you for the likes. That's really kind of you. Right, here we go. Let's go for a bit of gene regulation. So okay, this is where it's going to be annoying. Let me move it down just slightly. This is because I've been doing things. I'm doing things differently. I need to put my logo on this on my slides as well. Is OCR a nice exam board for biology? I like it, but it really just depends what type of student you are, whether or not you find the way they ask the questions good or not. It's a difficult one to be sure of. Um, do you know, it's a funny one. So Paul's mentioned about freshers uni blast from the past. I actually was, obviously I went to uni as a fresher, but when I went as a fresher, I wasn't really doing the fresher thing because I went as a mature student and I was already working in a nightclub at that point. So I was already doing like night shifts at the club. And um when it came to my second year of uni, I was part of the freshers team that got everybody out playing the games and stuff like that. And I love seeing the pictures of those days when we would play like the games in the union and stuff. It was lots of fun. I did uni was a place where I had lots of fun, but also discovered my joy for learning in a sense. So I do love my uni years when I look back at them. Anyway, right, Stephanie, here we go. Here we go for you. Right, regulation of gene expression. So you should know from module 2, transcription and translation. Transcription, we're copying the mRNA. Because we're copying the mRNA, oh my word, no, we're not. We're copying the DNA code onto mRNA. Be very specific in what you're saying. Because we know transcription happens, we've got our helicase that's going to unzip. We've got our RNA polymerase that's going to build our phosphodiester bonds. We know that we have got our pre-mRNA that's going to be made because of complementary base pairing, because of our nitrogenous bases. So these are all module 2 knowledge that needs to come through. Then what we need to do is we need to now talk about, well, how can we regulate the expression of a gene? So when genes are expressed, they are, if they're coding, they are going to code for a protein to be produced. So if we're going to regulate that gene expression at a transcriptional level, we're talking about the gene being turned on or the gene being turned off. So if we're going to transcribe it, it's going to be on. If we're going to inhibit that transcription, it's going to be switched off. When cells differentiate, they switch genes off. So when you start off, excuse me, when you start off, what you have is all your genes are on, and then as cells start to differentiate, they start to switch off genes. So that's something that we know, that's something that we talk about, and we always mention that. But what's important is understanding that actually sometimes hormones, the external environment, can actually inhibit or promote transcription from occurring. So I'm all naturally grey right now. I used to have jet black hair, like so jet black, it was ridiculous. And now, like you can see, like if I pull this bits back, you can clearly see all these bits where there is no pigment being produced in particular areas. So you've also got that as you get older, there is the switching on and switching off of genes. So Paul, you mentioned about the fact that you're totally bald, so you don't have the hair being produced anymore. Whereas for me, it's about the pigments in specific areas. So this regulation of genes. We know a gene is a section of DNA. We know that it is completely, the complete sequence is going to code for that entire protein. And going back to module 2 again, you need to understand module 2 that you've got your primary sequence. Your primary structure is that sequence of amino acids, and how that sequence of amino acids come together is determined by the DNA code. Yeah, it is to do with the gene. So I started going grey at the age of 17. So some forms of things like baldness or greying or pigmentation in different areas, that is because of the proteins or lack of proteins that are being produced. So when we talk about regulating this gene expression, one of the key ones in eukaryotic organisms is that you should know during interphase that your DNA is loosely wound. And if your DNA is loosely wound around the histone proteins, remember, histone proteins are positively charged. The DNA is negatively charged. Therefore, they are attracted to each other. And when they're together, the histone protein and with the DNA wrapped around twice, that's known as a nucleosome. So we can regulate transcription in eukaryotes by having them tightly wrapped around histone proteins or loosely wrapped around histone proteins. So a nucleosome is a way in which we are going to regulate transcription. We're doing this at the transcriptional level. If we are either allowing or inhibiting transcription to happen, that is at the transcriptional level. So interphase, you should know G1, SG2. G1 is the point in interphase where most of the protein synthesis occurs. Well, if protein synthesis is occurring, it has to occur through the first stage, which is transcription. So therefore, transcription has to, yes. So when we look at nucleosome, it's in a sense, a transcription factor is something that is either going to inhibit or promote transcription from happening. So in this case, with a nucleosome, we're regulating that transcription. Now, transcription factor is just the key word that OCR want you to use. Give me a second, I just need to move this up. And we use the term transcription factor for anything that can help either inhibit it or promote it from happening. So acetylation, for example, excuse me, I've got a bit of a runny nose tonight. Acetylation, these again are just examples to make it make sense. I feel that sometimes when we say, ooh, transcriptional level, you need to know about transcription factors. If no one actually describes what that transcription factor is, it doesn't make sense. It's just like a word, a different language. So a transcription factor would be something, for example, that is going to add an acetyl group to a histone, reduce that positive charge, therefore the DNA is going to coil less tightly. Now RNA polymerase can get in and access the promoter region. So a transcription factor is going to expose the promoter region so that the RNA polymerase can bind to the promoter region. So that's one of the things to think about. I talked about interphase and it being loosely wound. You know, during prophase, the heterochromatin, what you've got is this condensed chromosome. So during prophase, whether we're talking about prophase 1 in meiosis or whether we're talking about prophase in mitosis, you know that your chromosomes condense, they're tightly wound around the DNA. So it depends with the transcription factor that's going to inhibit. It might be that it blocks it. So you could talk about it being a repressor protein. You could talk about it as being a molecule that's going to bind and prevent the RNA polymerase from binding by blocking it, or it's going to bind to something else which is then going to inhibit the RNA polymerase. So if you think about RNA polymerase is an enzyme which can have a competitive inhibitor or a non-competitive inhibitor. So it really depends which one you're talking about. As long as you understand, it's either going to promote the binding of RNA polymerase or hinder the binding of RNA polymerase. That's the key part about a transcription factor. It helps it or it stops it. That's basically what they're looking for. When it prohibits, it really depends. I mean, most of the times we tend to say it's a repressor protein, but it may actually be a inhibitor molecule that would bind to an allosteric site. So it really depends on what they give you as an example. And that is all dependent on what they decide is the question in the context they want you to apply it to. So it's about you understanding that it's all about enzymes. Enzymes are all about proteins. Proteins are all about their structure. So the primary sequence that codes for the or determines the tertiary structure that gives it its shape, that allows it to function as it needs to be. So if you have a transcription factor and that transcription factor is going to promote transcription, it will therefore bind and change the tertiary structure, allowing for the RNA polymerase to bind, for example. No. Stephanie, you don't have to know these, but what they will do is potentially in a question give you this information and expect you to then apply your knowledge. So if they talk about methylation, and they have done that in the past, or acetylation or phosphorylation, do you understand if you've got an enzyme and they describe with that enzyme's name, whatever that may be, and they say it's going to do acetylation, they would say acetylation is the adding of a acetyl group, reducing the positive charge. And they then want you to explain to them that because the DNA is coiled less tightly, because they've given that in the stem of the question, that allows RNA polymerase to be able to do transcription to occur. So it's you applying your knowledge. But yeah, if you're going into acetylation and methylation, it depends, although Paul, it does depend on the exam boards. Some exam boards do expect them to know acetylation, methylation. OCR don't expect it, but what they do want you to be able to do is to put across the knowledge that you have to the stem of the question, whatever the stem is. And so, yeah, it really just depends. IB, you have to know methylation, acetylation. OCR, you don't. But they do use it sometimes, which is why I never don't give it whenever I'm doing my sessions. Sometimes going into a bit more depth also helps it to make sense, I think. Yeah, exactly. So a lot of the time with the application questions, and this is the thing that most students found this year, they had gone through and memorized a lot of the content. And so then they were like, oh yeah, I'm ready for this exam. I've got it, I can do it. And they went in and the stuff they'd memorized was given to them on a plate in the exam stem of the question. They were literally like, it's all I knew. What is going on? And because they'd memorized the content and the question then asked them to apply it, they were like, yeah, don't know what to do here. And so they got completely stumped. The important thing to note is whatever they're throwing at you, you've got to take a step back and say, okay, where does this lie? It's not an easy process to do. But again, if they're going to ask a question like DNA demethylation, so they would expect you to know removal of a methyl group. So there will be, for example, a picture that shows that if they're going to do anything on that type of question. And again, I have seen a question where they've done this, which is why I like to show it. So here you have cytosine. Cytosine, we know, is a nitrogenous base. So because we know it's a nitrogenous base, we know that if, in this case, they tell you a bit of information, if we have added a methyl group to it, it's now methylcytosine, and that therefore is going to inhibit transcription because it's going to inhibit RNA polymerase from having access to it. So, Stephanie, to answer your question for OCR, what do you need to know about transcription factors? That they can either promote it, allow it to happen, or inhibit it, prevent it from happening. And this is at the transcriptional level. We are either promoting transcription or we're inhibiting it, depending on the example that they've given. And it's their choice which way they go. on that one. Cool. Right. Okay, so transcription, we know, copy the gene, that DNA sequence into mRNA, DNA helicase unzips, free RNA nucleotides, complementary base pairs, RNA polymerase, building phosphodiester bonds, pre-mRNA. These things should come to mind, anything to do with transcription. So you should know that if we have unzipped, we've broken those hydrogen bonds, if we've had the free nucleotides come along and we've then built that polymer because we have joined the nucleotides together through this phosphodiester bond, then what we end up with is pre-mRNA. That is just module 2 knowledge. Now to take it up to module 6 knowledge, what you have to understand, this is really frustrating now. I'm going to bring this down here. 1 second. There we go. Still at the post, this time going slightly past transcription, we've transcribed. So there we have our RNA polymerase binding at the promoter region. We've transcribed and we've got this pre-mRNA. This is where your exons and introns come in. So you have coding regions and non-coding regions on your chromosome, because remember, a chromosome is made up of lots and lots of genes. Some of them are coding regions, genes. Yep, go for it, Stephanie. Ask away. This is the whole point of why I do these sessions, by the way. I do them so you can ask the questions. I could drone on, you can watch my videos on them. Do they need to know sense and nonsense? Yes, anti-sense strand, not nonsense. It's called sense and anti-sense strand. Although technically they won't lose the marking point as long as they say they use the template strand, but yeah, they should know the sense and anti-sense strand. Oh, Angelo, thank you for that. I'm really pleased that you passed. Well done. Congratulations. Okay, hydrogen bonds are broken by DNA helicase. Yes. One DNA strand acts as template. Yes. Free RNA nucleotides bind to complementary base pairs. Yes. Uracil replaces thymine. Yes. RNA polymerase joins the nucleotides with phosphodiester bonds. Yes. Perfect. Is this the correct description of transcription? Yes, the only thing I would say that you need to add on is the phosphodiester bonds and also to produce pre-mRNA. Oh, Angelo, thank you for the rose. The 2 roses, actually. Thank you. That's lovely. And this is the one in my notes. Do you think I'm missing any key? Any key points? Not for transcription, no. I think that's fine. Just add in a few extra bits. Yeah, actually, Paul's right. mRNA being the single-stranded, and the fact that it's single-stranded and short allows it to go through the nuclear pore. Oh, Angelo, that's amazing. Do you know what? I wish you could pay me millions too. I actually, do you know what is really exciting? This is really sad, but it's really exciting. I have made it on YouTube too, that my videos, I now, if people watch videos, annoyingly, they have to watch some ads, but I actually earn on that. I mean, don't earn a lot, but still, now that people, when they watch my videos, I can actually earn on them. So that really helps. But yeah, I wish you could pay me millions too. Do I prefer AQA or OCR? Oh, thank you. I don't prefer either or. I like both of them separately because actually they both have different unique. I tutor, interestingly, more AQA students are who I tutor now compared to OCR privately. I've got obviously my group tuition sessions and a piece of time on course. I wish I'm barely making minimum to be able to actually cover my wages for the year. It's a funny one. I worked my way up to being assistant principal, had the girls and everything. Everything just changed, and now I'm happy that I get to take them. We did gymnastics tonight before I came out here to work for my group tuition sessions. So currently I'm still building up my clientele schedule at the moment. But yeah, I wish the pizzas were on me. Um, I wish that was the case. I'm hopeful. I currently can't even afford a coffee since I've been able to earn on YouTube for what, since the 15th, almost 15 days, almost 15 days. And at the moment, I've managed to make just over £3, but I don't get any of that. I don't see any of it until you reach £60 minimum. But yeah. Although anyone who watched my videos, that's awesome. That it means that I can now get a little bit payback. A year's worth of work and the hours I put into it has made a difference. But the key thing for me is that I want to help people. I want to help students. I'm worried at the state of education at the moment. I'm worried at the situation that schools are finding themselves, that the qualified teachers are leaving the profession because it's just not fulfilling anymore. And so therefore the new teachers that are coming in, and I remember what it was like being a new teacher, are coming in and scrabbling their way through trying to learn the content that they have to teach without having the experienced teacher there to help them and guide them on how to show students how to learn. And that's one of the other things as well, is that I have got so many teachers that I've worked with in the past that really helped me, and that made me a better teacher. And then I became the teacher who was training the other teachers so that they could teach the content. And then, yeah, it just changes. And so it's a hard one. It's a hard situation, but I hope that my resources help students enough. And that's getting students email me to say, this is amazing. Thank you so much. It now makes sense. And also for me, because sometimes I forget. I'm like, I haven't taught that in ages. I must go back and refresh my mind on that. Having ADHD, it's a wonderful thing, but it can also be a hindrance when you are forgetful. So I have to keep myself fresh, and also I want to keep myself fresh and learn more. So I've been doing a lot on cereal dilutions. So I've got some videos coming on cereal dilutions when I get around to finishing them. Yes, I do, Stephanie. I have on a Monday evening. I don't have one next week. I'm having a break because I've got the girls whilst Alan is away. So I do normally on a Monday evening at 7:30. That's why I'm on here, because my current video is downloading for me to email it to them. I do that for A2 content, although we're covering AS at the moment. And on a Wednesday, I've got year 12 for AS content. So depending on which year you are, obviously, I'm guessing you are year 13 because you're talking about 6.1.1. So yeah, if you are thinking about group tuition, it's on a Monday. It's £25, and there at the moment there are 3 students in the class. So it's a small class, and it's very interactive, and it's also where I assess your written ability and feedback instantaneously after the sessions. Angelo, have a great evening yourself. Oh, thank you. Oh, thank you. That's really lovely to hear. Thank you so much for that. It is lovely to hear that. Okay, back to this. So, and obviously in my group tuition sessions that I do, I plan them meticulously. This isn't just me going blah, 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 and chatting away. Those ones are planned because they are paid sessions. These ones, I'm just kind of like, hi guys, I'm here. Come watch my videos. So yeah, I want to help as much as I can for those of you who have questions. But back to this one. What's really interesting about this part, the post-transcriptional level, so we've transcribed, we've copied that, like you described earlier, we've copied that code onto the mRNA, but we have some bits in there that are non-coding regions. And so therefore, what's going to happen is we're going to splice them out. Now, you don't have to understand how the splicing works, but basically you have these things called spliceosomes, these complexes that come along, and what they'll do is they will slice and re-stick together the exons And what that means is you end up with what's known as mature mRNA. So pre-mRNA before splicing, mature mRNA after splicing. So we're still in the nucleus at this point. We haven't yet left the nucleus. So it's post-transcriptional because we are still modifying that mRNA that's been produced. And so what you've got then is the splicing out of those introns means you could end up with a mature mRNA, which is exon 1, 2, 3, 4, and 5 in that order. Or it could be that it's exon 1, 2, 4, 5. Or it's exon 1, 3, 4, 5. Or it's exon 1, 3, 5. Or it's like a whole random combination of it. Why is that important? Well, the reason that's important is remember that code, that transcriptional code, that is the one that is going to be translated later on. And that translation is going to be putting in the right sequence the amino acids that make up the chain for the polypeptide. So if I'm shuffling these exons around, what it means is I can make, and it tells you up at the top, post-transcriptional level mRNA modification to regulate translation and the types of proteins that are produced. So if I've got different exons being brought together, I'm going to have different sequences. Those different sequences are therefore going to code for different sequences of amino acids. So all of that is a way in which we are regulating gene expression. We've done the transcriptional level, switching on and off of the genes by either promoting or inhibiting RNA polymerase. Now at post-transcriptional level, we are splicing out those introns. Therefore, we are modifying the mRNA to give us that mature mRNA. Oh, and also we add a cap and a tail to it. And that's so it will not be broken down as it gets to the ribosome. So you've got to remember that the mature mRNA has to leave through the nuclear pore and make its way and bind to the ribosome and bind to the small subunit of the ribosome so that you actually have the whole ribosome coming together with the tRNA and the large subunit and that building process occurring. But we need to transport it there first, and actually it can be encountered by, let me think this one through, enzymes that might actually break it apart. So by adding a cap and a tail to the end, what that then does is that then helps it to not be broken down, so prevents degradation and aids it binding to the ribosome at the right place. So pre-mRNA, introns are spliced out. We've now got the mature mRNA, and that mature mRNA is the bit that leaves through the nuclear pore. So this is why it's important to know your Module 2 content, but then take it that step further to the Module 6. And then what you get is translation occurring. Obviously, translation is going to happen at the ribosome. We need to remind ourselves that it's about amino acids and the fact that there are 20 different amino acids. So what you have is those 20 amino acids, all different because of that R group. And they come together through, oh, that's annoying. Let me move that down. mRNA moves to the ribosome. I should actually, let me just add in, that one should say mature mRNA, because we just discussed that now after post-transcriptional level, it's now mature mRNA. And then you have the tRNA molecule. So you should know that the tRNA molecule is a polynucleotide. So tRNA is a polynucleotide. A nucleotide obviously is made up of a nitrogenous base, of a ribose sugar here because it's RNA, and a phosphate. So what you can see is this clover leaf shape is because of interactions between nitrogenous bases, and it always has the same shape. And this bottom region here is the area which is known as the anticodon, and that will complement the codon, which is found on the mRNA. And then that brings along with it the corresponding amino acid. Now, they don't expect you to know how the amino acid and the tRNA molecule come together, but actually they could ask a question about it, because they could say, well, there's an enzyme that ensures that the corresponding amino acid is joined with the tRNA molecule. And so therefore they could ask you about the fact that the enzyme would have to have the active site that was complementary to joining those two things together. So what you have to remember is knowing the content is only so good if you can then apply the content to a completely random situation that they may give you in the exam. And that's the bit that most students struggle with. They struggle with understanding how what they've learned in their lessons relates to that random question that somebody's come up with in the actual exam. Obviously, we have our peptide bonds forming because we've got two amino acids. So between the carboxyl group on one amino acid and the amine group on another, we have a condensation reaction because we end up with water as a product too. And then you've got your primary structure of your polypeptide, and that will then lead to your secondary and tertiary structure, which obviously will then give you, it's really annoying that it's doing that, the polypeptide, which is in this case a globular protein. Moves, can I say attaches? Yes. Wait, well, hold on a second. I've said yes. When I say moves, where have I said moves? I may have said moves in when I was talking about it. What do you mean? Can you say attaches? I said it moves from the nucleus through the pores to the ribosome. So you would want to say that it moves from the nucleus into the cytoplasm and then attaches to the ribosome. Well, what are you saying parrot for? You've now confused me. I'm like, what does that mean? I don't know whether there is. Oh, hold on. I missed that one before. Oh, yeah. Understanding is so much more important than parroting. I did see that. Yeah, so they don't need to know. Interestingly, they don't need to know about the EPA sites for OCR or even AQA. IB they used to have to know it, but they took it out. Yes, so the mature mRNA is going to attach. Oh, I see where it was above. I couldn't see it. Moves to and attaches. Yeah, so you could and attaches to the ribosome. Interestingly, a lot of the bullet points for this kind of thing come straight from the exam board. Yeah, they don't need to know about the EPA site. And it's funny because I always call the EPA site the exit polypeptide amino acid. That's how I remember it. I know it's not. But that helped me to remember the E site was the where the tRNA will exit. The P site is where the polypeptide is produced, and then the A site is the site where the new amino acid comes in. But yeah, no, they don't need to know that at all. Okay, so the last thing I'll finish on because it's getting close to my bedtime. I say my bedtime. I get off of here and then I do my scrolling. I'm like, stop scrolling. Or I listen to my book. I just finished a good book actually. Atmosphere. There we go. Right. Okay. So the last one you have to know about, they took out the translational level. They removed that. And now you only have to know the three. So you have to know transcriptional level, you have to know post-transcriptional level, and you have to know post-translational level. So transcription, We are RNA polymerase, post-transcription, mRNA being modified, post-translational level. So we've already had translation happen. We've built our primary structure, and that primary structure is then turned into the secondary, the tertiary structure. Now at post-translational level, this is where the proteins can actually be modified after translation. So this is where you have things like adenylate cyclase is an interesting one. Protein kinases, another interesting one. So these are proteins that actually are inactive but will be activated when signaled to do so in the cell or in the body if they, depending on what is needed. So in this case, we know that I have got this inactive protein kinase. I've got a protein which is an enzyme which is inactive, which has the wrong active site shape. So what then happens is, for example, cyclic AMP, which is produced after adenylate cyclase has been activated. So adenylate cyclase is activated. That will therefore convert ATP into cyclic AMP. Cyclic AMP can then bind to the inactive protein kinase, activating the protein kinase. The protein kinase is then able to then go on and do whatever is needed within the cell. So it could be, for example, adrenaline binding, the protein kinase is activated. Therefore, we now have enzymes to break down glycogen into glucose so that we have more glucose molecules available. So hydrolysis is occurring, glycogenolysis. That could be one of the things. It could be the ADH, antidiuretic hormone, has bound, and it has bound to the collecting duct cell. When it binds the collecting duct cell, that in itself activates the ATP to be converted into cyclic AMP. Cyclic AMP activates the protein kinases. That therefore means more aquaporins go into the plasma membrane, which therefore means reabsorption occurs of water. So when glucose is high, cyclic AMP is low, isn't it? Why? So Stephanie, my question to you would be, you want me to confirm to you that you're right in what you said, but I would say, why would that be the case? When glucose is high, cyclic AMP is low. Why would that be the case? Yes, post-translational is the only one you need to know, although the textbooks still have the translational, but it's just the post one. And I say refer back to the specification always when you're doing your revision. But yeah, Stephanie, I've come back to you and said, why do you think when glucose is high? I'm not saying you're wrong. I've said you're right or wrong here. I want you to explain why when glucose is high would cyclic AMP be low, because no need to produce. Okay, so what do you, that's not connected to it. So what cyclic AMP, which is why I asked the question, and this is the important thing of understanding your different molecules. And so therefore, I would say your reviewing of nucleic acid nucleotides would be important to make sure you feel confident. Give me 1 second. Let me just get my slide up. Oh, no, that's not the one I wanted. It's this one. Where are you? Where are you? Where are you? Oh, maybe it's not that one. I don't know where I put it. Give me a second. I'm going to move this across. Okay, so here's ATP. Oh, my phone's telling me it's time for bed. Here's ATP, here's ADP. So ATP, adenosine triphosphate, ADP, adenosine diphosphate. And if I were to bring up adenosine monophosphate, as you just saw a minute ago. That one, I'll bring that in here. And I will remove its animation. There we go. So here is cyclic AMP. Try that again. I can't remember. I need to revise it. I think. So if you have, and this is the interesting part, and this is important in ensuring you know where to do the revision. So that is a good thing that you asked the question, and it's a good thing that you actually tried to answer it yourself, which why I push back, and it's how I do my sessions. Because a lot of the time you're like, yep, I know it. And then you get asked a question a different way, and you're like, oh, actually, I don't know this. I need to go and revise that. So that's the important part. So obviously adenosine triphosphate, where you have 1, 2, 3, adenosine diphosphate, 1, 2, adenosine AMP, the M stands for monophosphate. So there's just one phosphate. So if we're going to go from ATP to cyclic AMP, we are therefore removing 2 of the phosphates. If there is what you suggested, if there is high glucose, if you've got a high glucose concentration, you would expect that glucose to be used in glycolysis. And if it's used in glycolysis, that would therefore mean that it would then go from glycolysis through to the link reaction. It would then go to the Krebs cycle. It would then go to oxidative phosphorylation. If there is oxygen present, obviously, because we're talking about aerobic respiration, and if that is the case, we would expect there to be lots of ATP molecules being produced. So when you have high glucose, you would expect aerobic respiration in the presence of oxygen, and that would produce lots of ATP molecules. Now, ATP is normally hydrolyzed down into ADP, and then ADP is going to be turned back into ATP through the adding of a phosphate at oxidative phosphorylation. Cyclic AMP is only going to be produced when it's activated. The adenylate cyclase, the enzyme, is activated for the production of cyclic AMP, because that now has a specific role to play in, for example, aquaporin's being added, or in the production of enzymes to break down the glycogen into glucose. So you're absolutely right in what you originally said. When glucose is high, cyclic AMP would be low. Absolutely, because if your glucose is high, you don't want to activate protein kinases to break down glycogen into glucose. And therefore, you wouldn't want both of them to be activated at the same time. Hence why it's a post-translational gene regulation. And I hope that's been helpful. I am going to stop there for tonight. I will be back live on, excuse me, on Wednesday. So do please pop over on Wednesday. And if you are interested in joining obviously my group tuition sessions on Monday, our year 13, if you are looking for recapping just AS content, then I am doing the Wednesday with my year 12 students. But yeah, please do check out my videos as well, because they are there to get you through the whole course. The whole course is on my YouTube channel for you. You are very welcome. The other thing I would say, Stephanie, to check out is aiqchat.com, which is the website that at 9 o'clock on Wednesday, Paul. I go live at 9 o'clock because I've got back-to-backs of students and my group tuition on Wednesday, and I need a little bit of time to do the downloading, etc. before I go live. So 9 o'clock on a Wednesday. But yeah, aiqchat.com is also a good revision platform because it will help you to identify where your gaps are. And we dropped the cost down to £5.99 a month. So it's, if you sign up, you will get a week's free trial to see if it's useful for you. The best thing to do with it is to use the AI tutor. It basically is like having access to me in your pocket. But anyway, have a good evening. Thank you for being with me, and I will see you guys on Wednesday. Bye, everyone.